Well, good morning, folks. Welcome to Neuro3301 yet again. Here we are. It's a beautiful fall morning. We've had a wonderful weekend of great weather, and we're all just really excited to get back to school. So, without any further ramblings and rantings and delay, let's get into the good stuff, folks. Let's get into the content. I know you're all screaming for that content. You're all saying, please give us the content. Give us the content. And I'm here. I'm just trying to dish it out as fast as I can. So let's get into it. Today we'll be talking about schizophrenia. And this is the first of a two-part series of lectures. The first part is today, and the next part is on Wednesday. And then, of course, we get a nice little fall break where we can recharge our batteries or catch up on reading or build the remainder of this course so that I don't run out of lectures before the year ends. Whatever it is that we need to do, we'll be able to get it done at this point. So let's take a look first at the assignments, because we've got the assignments graded. If you haven't checked yet, you can visit Brightspace and you should be able to see your assignment. The TAs did a great job with grading them, so you should be able to see per question feedback for the assignment. You could see, on a mark-by-mark basis where you lost points and maybe a brief description of why it was that you lost those points. The class did very, very well overall. So the class average was 87 and 1/3%. So that's a nice, juicy, high average. We love to see it, folks. We love to see it. The median is 90%, meaning that half the people in this class got over 90%, which again, we love to see it, folks. We love to see it. Don't you love it? That's great news because this is kind of the whole sort of aim that I have with the assignments, is that they're somewhat deterministic. They're almost more of like an exercise that takes you through in a step-by-step way a certain pathway or a certain set of approaches to doing an analysis, and you follow the steps and you watch the video and you learn something about how the analysis is done and about how the tools are used, and for your efforts you get some points. So this is kind of the goal, and that's not to suggest that every assignment is going to be easy or straightforward or simple, but this is kind of the overall approach that I'm trying to take with these. And hopefully, if you like I say, if you were disappointed with your midterm grade, this would hopefully be a bit of welcome news that you're not that you're actually going to probably just do just fine in this course if you put in the effort and the work. Because the material is varied and there's lots of opportunities there to earn points. So good work, everyone. Thanks to the TAs for your great work in grading, and congratulations to anyone who got a mark they're happy with. And if you're not happy with your mark, it's of course an opportunity to improve. Contact your TA or come to my office hours if you have any questions about any of that kind of stuff. So let's get into the good old content. We're going to lower the elevator into the content mines and start dredging up some of the good old content that you guys know and love and crave. So we're going to talk about schizophrenia in a little bit more detail than the other topics. And part of the reason for this is, I think when we start getting into the swing of things with covering disorders, what you'll probably find is that many disorders and many traits and conditions have a similar pattern to them in terms of what we know. So for most conditions, there'll be at least one GWAS study, perhaps several. There'll be some epigenetic studies, and there'll be some other kinds of studies as well that look at developmental programming and so forth. And I think it can become a little bit repetitive to go through every single topic in that level of detail, because you start to learn the pattern. And really, frankly, when you're thinking about GWAS results, oftentimes there's not a lot of specific insight to be gained. You might discover that a given condition has 35 SNPs associated with it, but the SNPs are in all kinds of random places in the genome that are very difficult to interpret. So I thought I'd load up the schizophrenia lecture with a lot of varied material to show you sort of the span of what could be covered and what you can learn about a given disease or a given disorder. And then the subsequent ones, oftentimes we won't go into every possible angle. I'll show you the parts of it that I think are the most interesting or maybe the most novel or informative, while you would have the knowledge that in the background, most likely the literature also has a lot of data that's this sort of the same kind of stuff that we're used to. So maybe not, so for every condition, I'm not going to show you a GWAS study and a brain DNA methylation study. I'm going to try to show you new and interesting things for every condition. But the schizophrenia lecture, I think, sort of sets the model for what things can be done. Schizophrenia, of course, is also a very serious and disturbing psychotic disorder, right? So this is a disorder that causes a loss of contact with reality, and in North America, it affects about 1 in 100 people, or about 1%. And not that I think diseases should be really, categorized on the basis of their cost. Probably what's more important is the suffering that they cause, not so much the money. But it does cost a fair bit. The loss in productivity and the cost of treatment adds up to about $60 billion in the US per year, and probably a correspondingly high amount in other countries as well. The prevalence of schizophrenia is fairly similar around the world. It does seem as though there are variations on like a continent by continent level, meaning that the prevalence isn't completely identical worldwide. Although, admittedly, it becomes very complicated to try to define schizophrenia in a reliably cross-cultural way, because it's not a disease where you can take a blood test and find out that you've got high levels of some kind of hormone that causes it. It's a multifactorial condition with many different aspects to the phenotype, all of which or many of which, I should say, depend on fairly subjective judgments of people's personalities and behaviors and their self-reported symptoms and so forth. As you would likely know if you took any of the other classes in this department, there are positive and negative symptoms of schizophrenia. The positive symptoms are essentially any symptom where you have a experience where you have a symptom that goes beyond the normal parameters of human experience, things like hallucinations and paranoia. Those are things that the disease adds to your subjective experience. And then there are, of course, negative symptoms where the disorder takes things away. So you have social withdrawal or apathy or a poverty of speech or a flat affect and things along those lines. Now, it's a disease that tends to be diagnosed in the kind of late teenage, early adulthood years. So a big question that people ask about this then is this something that you're sort of born with but doesn't manifest until later on in your life? Or is it sort of like an injury that happens to you that could have easily been, or could have somehow been averted had something gone differently for you in that period of your life? That's a very tough question to answer. About 85% of the cases of schizophrenia have what they call a prodromal phase, where the person would not meet the diagnostic criteria for full-blown schizophrenia. They wouldn't have the florid psychosis and the massive social withdrawal and all the other kinds of symptoms, but they would have what you might describe as odd patterns of thinking, perhaps some level of apathy or social withdrawal, symptoms that in retrospect seem very indicative of a future diagnosis, but at the time you could write off as something else. Oftentimes, I think people kind of who are on their way to on their way to schizophrenia, display symptoms that many would describe as like depression or anxiety or some other sort of lower grade mood disorder. I think partly because there's a sense of optimism where, they couldn't possibly have something as bad as schizophrenia. It must just be depression. If we could just get them, if we could get them into some more sunlight and put them in on a sports team or something, then maybe that would fix everything. It's difficult to tell, but anyway, at any rate, you can go back through patient histories and find that very often people who go on to develop schizophrenia have behavioral markers ahead of that time. The age of onset tends to be sort of peaking around age 20, I think, in males, and the age of onset is somewhat later in females, coming more toward the later 20s. And it remains an open question as to whether this is because brain development plays out differently in the two sexes, or if there's some difference in the way the symptoms manifest socially, the way that the social environment interacts with symptom presentation. But it's important to note that in both cases, the majority of diagnoses are made fairly early in life, and it's quite rare to receive a diagnosis much later than that. And those who do there are often other reasons for it, potentially just something as simple as they had been misdiagnosed as something else in the first place. Like, for example, bipolar disorder can often have psychotic manifestations, and so people can be diagnosed with borderline personality disorder, or sorry, not borderline, bipolar disorder, and only later on do people realize that schizophrenia might have been a better diagnostic choice. But after diagnosis, there's a decline in cognitive and social function that, if the person survives long enough, will eventually sort of level off, leaving the person in a comparatively worse off state, but without any subsequent decline. So there is certainly the possibility of learning to live with this condition if you can. The treatment options remain limited, so people are very curious about this, because antipsychotic drugs, of course, can treat the positive symptoms, but they don't really do a whole lot for the negative symptoms. They're also very unpleasant to take. They have a number of really pretty awful side effects, and so for perhaps good reason, it's difficult to maintain patient compliance. People oftentimes will go off their meds because the effects that they cause on the brain and on the body are really just too gruesome to bear. Now, it's been known for quite a while that there is a genetic link to schizophrenia. You can look at the relative risk of developing schizophrenia given a diagnosis somewhere else in your family. So here is a plot from a abnormal psychology textbook showing your likelihood of developing schizophrenia, assuming somebody else in your family has it. If you're a monozygotic twin and your other twin develops it, you basically have a 50-50 chance of yourself developing schizophrenia, close to 50-50. If both of your parents have schizophrenia, then your odds are also very, very high, a little bit higher than 45%. If you have a dizygotic twin with schizophrenia, your odds go down to around 20%. Of course, the baseline risk is about 1%, so 20% is many times higher than what you would be or the risk you would have if you were just a random person on the street. And you can kind of go on down the line. If you have a child who has schizophrenia, your odds of yourself having it are somewhere between 10 and 15%, and so on down the line. So even if you get as far as grandparents, or if you're a person who has grandchildren with schizophrenia, your odds yourself of having them are around 5%. I don't know exactly why they structured it this way. It would make more sense to say if you were a grandchild of somebody with schizophrenia, but the relationship is kind of the same in either direction. At any rate, this tendency to run in families does seem to clearly point toward a genetic cause. That, like I said, the concordance in monozygotic twins is about 48%, and in dizygotic twins, the concordance is 22%. You could actually go back to our lecture on behavioral, or whatever it's called, the thing where I talked about the twin studies anyway. You could take those figures and plug them in, and you could probably get an estimate of what the actual heritability would be. I haven't calculated that here. But anyway, it's suffice it to say there is clearly a genetic link somewhere. And really, whenever you have a condition that's as serious and debilitating as schizophrenia happens to be, I think it's natural to sort of reach for a genetic explanation, because it just seems that something so debilitating and so severe couldn't possibly just be because you ate you ate a bad plum or something, like some kind of event happened in your life that brought you into this long-term and essentially irrevocable state of psychosis. There must be something biological happening at a deeper level. Although that's not always that's not the only viewpoint. There are people out there who would argue that schizophrenia is purely a cultural creation, and that it's something that may not have been seen as pathological at all in different cultures or at different times in human history. So, like Thomas Szasz and the whole myth of mental illness thing, people will come up with that argument, but I think it's becoming less and less tenable now that we have so much biological data out there. I think when I was in undergrad and taking psychology, I read Thomas Szasz's book about the myth of mental illness. He was sort of a kind of a libertarian critic of the psychiatry industry. Himself, he was a psychiatrist, and this was a very fashionable thing in a way. And I think it sort of reached a cultural moment during the Bush era Scientology boom, when everyone started talking about Scientology, and Tom Cruise went on Oprah and was jumping on the couch and berating people for going to psychiatrists and whatnot. There was a big moment there where anti-psychiatry was culturally quite relevant. It eventually fades away, like anything does, when the weight of evidence accumulates and it becomes obvious that like these anti-psychiatry types only have one argument to be made. Meanwhile, the real world of science is continually accumulating new data. You know, it's easy enough to argue that schizophrenia is fake in the 70s when there are no genetics and there are no, you know, the neuroscience that we have to work with is very, very limited, and you can point to a lot of unknowns and say, well, maybe it's all happening in this unknown area. But as our knowledge of biology and neuroscience and genetics improves, the room you have to hide your lack of knowledge becomes smaller and smaller. The amount that's unexplained becomes smaller and smaller, and it becomes less and less tenable to argue that it's all just a big lie. You know, the same thing actually happened with creationists back in that sort of early 2000s era as well. It'll be interesting to see what happens to the modern day skeptics. I guess skepticism is always warranted, but science will continue to march on whether or not people believe in it. Ultimately, reality asserts itself, and if you're right, you're right. You can show it eventually. Anyway, sorry for that diversion. I just think having kind of been following this stuff long enough to note the major changes in culture, it's fun to look back on it and see who was right and who was wrong in all these arguments. So let's get into the genetics of schizophrenia, and today I'm going to give you a historical approach. I won't always do this because we have 2 lectures to talk about schizophrenia. I have a little bit of space to stretch out and give you kind of a historical sense of how the genetics of schizophrenia was addressed early on in the pre-genomics era, and contrast that with how it's addressed now in the genomics era. And after that, we won't really need to get back into this stuff too much now that we're in the genomics area. Everything that I tell you from here on out will be in the genomics world, and we won't need to worry about all these weird, old-fashioned kinds of studies. But for now, I think it's valuable to give you a survey of where we came from and let you contrast it with what we have now. So you can begin with the starting point that schizophrenia clearly has a genetic component. There's really no disputing that when you look at the data from twin studies and the data from pedigree studies and these kinds of things. However, the genome is huge, and you have a lot of places that you could look. Where in the genome are the causal variants that actually influence your risk of schizophrenia? Prior to the genomics era, when the genome was sequenced and you actually knew where everything was, it was a little bit like finding your way in the dark. Researchers had genetic maps. They had cytogenetics, where you could look at the different chromosomes and you could identify different bands, and you might be able to somewhat understand things in relationship to what arm of the chromosome they are on or what band within each arm they're located on. But all of this was very roughly defined. However, there were a few things where people were fairly confident about the location. And one of these things, they're called microsatellites, microsatellite markers. Polymorphic microsatellites, to be precise. So microsatellites are short stretches of highly repetitive DNA. These are also called short tandem repeats. They're bits of repetitive DNA. They look sort of like this. Repeats can be, between 2 or 3 or 4, or, within a small number of base pairs long. But that same base pair motif is going to repeat itself many, many times in a given repeat. And they are highly polymorphic. And what's polymorphic is not the motif of the repeat so much, but rather the length of the repeat. Polymorphic, I should point out, just means many forms. Poly meaning many and morph meaning form. These are old Greek words. So anything that's polymorphic differs considerably between people. SNPs, of course, are polymorphic. You can have multiple alleles of SNPs. But so are tandem repeat lengths or microsatellite repeat lengths. Part of the reason for this is simply because when you have a bit of repetitive DNA, it's very difficult to copy it faithfully, because the DNA replication machinery has a hard time working when it's sort of on a repeat. It's like if I were to ask you to recite a very repetitive poem, or I'm trying to think of an example here. Well, I don't know. It's like kind of if I were to make you try and recite the digits of pi, it might be, well, but then pi is kind of definitively not repetitive. Sorry, I'm trying to think of an analogy in real time. But anyway, if you're trying to recite something very repetitive, it's easy to lose your place. That's putting it in the most simple terms. And it turns out that this same flaw exists in the DNA replication and repair enzymes. So in areas where the genome is repetitive, like microsatellites, the length of these microsatellites can vary considerably between people, just on a matter of length. Maybe there's 10 copies of the repeat, maybe the other person has 20 copies. There can be very, very large variations. Now, this is quite an advantage when it comes to genotyping people, because SNPs really only have two forms. There's one allele and there's the other allele, and somebody could either have one or the other, or they could be heterozygous and have one of each. But that's actually not a huge amount of variation. Microsatellites, on the other hand, can come in all kinds of different lengths. You could have a length of 1 repeat, or you could have a length of 10 repeats, or you could have a length of 50 repeats, and everything else in between. So there's a lot more variation to work with there. It's a quantitative form of variation, not a qualitative form. So you can use these, if you happen to know where they're located, you can use them to genotype people and develop what's called a genetic fingerprint. So the way that works is with PCR. We've talked about PCR before, but imagine you've got two different microsatellite alleles. You've got somebody over here who has a short version, and then you have somebody over here who has a long version. The content of the repeats is the same. It's just ATC, 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 repeated endlessly. But the number of repeats that the two people have are different. Now, you can use PCR primers that bind to flanking areas. The PCR primers don't bind to the repeats themselves, because the repeats themselves are not specific to anywhere in the genome. They can show up anywhere, as you might have remembered from the first assignment that you just all got your marks back for. So the primers are binding to the flanking sequence, which is unique and is located in some specific known location in the genome. The primers bind to either end of the flanking sequence, and the PCR process will amplify whatever is in the genome that's between the two primers. That's the magic of PCR. So when you run people's samples through this process, you'll get PCR products, so you'll get amplicons that are of variable length. In person 1, you would get, whoever has allele A, that's a shorter allele. And when you look at the PCR products from their DNA on a gel, you use gel electrophoresis to sort out these PCR fragments by size, they would have a smaller fragment. On electrophoresis gels, the smaller bits of DNA run faster, and they run all the way to the end. Small things move faster, so they get further in the same amount of time. The larger fragments with higher molecular weight are bigger, and they move more slowly, so in the same amount of time running the gel, they move less far, so they're up toward the top. And you can essentially just look at the position the PCR products end up on this gel and genotype people on the basis of repeat length with that. You can use a ladder that tells you the length of the different parts of the gel, so there's ways to quantify it that I'm not showing here. But ultimately, you can determine the repeat length for any given microsatellite. And that repeat length turns out to be fairly heritable. So for most of your microsatellites, you would have repeat lengths that are fairly similar to what your parents gave you. Those are heritable just like everything else in your DNA. However, they're very polymorphic between groups of people, and nobody really has the exact same combination of microsatellite repeat lengths. So if you were to genotype hundreds of, 100 or so of these microsatellites spread throughout the chromosomes, what you'd find is that almost nobody has the very exact set of the very same exact fingerprint. People might have different, somebody might have a long version of, one microsatellite and a short version of the other. Other people might have the very exact opposite. So it's a way of uniquely identifying people without actually even getting into their SNPs or anything like that. This is commonly used back in the days, I think it's still used for forensics, because you can genotype people on repeat length polymorphisms and get a fingerprint that is almost certainly unique to one single person on this planet, except maybe them if they have a twin. A monozygotic twin, I should say. And then you can determine if a DNA sample is a match for somebody else without ever even having to sequence it, because the odds of two people who are not related having the exact same pattern of repeat length polymorphisms is astronomically low. So if you carry out this process and genotype the repeat lengths of a bunch of microsatellites, you'll have a signal that's very unique to each person, right? Okay. Why are they called microsatellites, by the way? Do they have anything to do with the satellites that are orbiting the Earth? No, but there's another interesting little bit of history there that I can't resist telling you. Back in the early days of molecular biology, people were arguing over where heritability comes from and what the molecule is, like what molecule actually carries the genetic information. And the contest was sort of between nuclear DNA and nuclear proteins. People knew, I guess, at some point that the nucleus of the cell is where the genetic material was, but of course the nucleus has DNA, but it also has histones, and those two things seem to be equally important. It's tough to tell what really has the magic spice. So the C value is simply a measurement of how much DNA exists in every cell's nucleus. I don't really know why it was called a C value. It's not a term that's used anymore. But it really concerned researchers because people can kind of begin with the premise that human beings are the fanciest and most sophisticated organisms on the planet, and therefore we must have the most complicated genetics. And if that's the case, we ought to have the most DNA. Like if DNA really is the molecule responsible for heritability, humans should have the most of it because we have the most stuff going on with our cells, and we have the most complicated. Well, it's assumed anyway that we would have the most complicated genome because we're the most complicated organisms. However, this is not the case. Amphibians, like salamanders and frogs, have about 20 times more DNA per nucleus than humans. And some kinds of amoeba have like 200 times more. Now, of course, you get into things like plants that can be, triploid or quadruploid or multiploidal, so they would have many copies of their chromosomes. That's kind of a different story. But there are some animals out there that just have straight up more DNA, even though the number of chromosomes they have is about the same as what we'd have. They'd still be diploid organisms. So how on earth could a salamander have more DNA than a human if DNA is supposed to be what's responsible for us being so special? This is a really fairly big question. Back in the 50s or the 60s, there wasn't really any gel electrophoresis going on, nor was there a whole lot of chromatography. So what people used to do to figure out the size of DNA fragments was use ultracentrifugation. So you would extract DNA from cells or from tissue, and then you would put it in an ultracentrifuge and run it against a cesium chloride gradient. Cesium chloride naturally forms a density gradient. So if you centrifuge stuff against this gradient, you essentially fill a tube up with this cesium chloride stuff, and then you put your DNA on top. The DNA, if you spin it fast enough, eventually the force of the acceleration will drive the DNA into the much denser cesium chloride. And what's interesting is that when this happens, the bits of DNA separate into bands based on their molecular weight. So the lightest stuff, or the stuff with the least amount of molecular weight, would end up near the top of the centrifuge tube, and other higher molecular weight things would get further and further down. This is sort of the same idea as if you fill a big box full of rocks and just start shaking it, all the heaviest rocks will eventually end up at the very bottom of the box, and all the sandy type stuff will kind of make its way to the top. Or is it the reverse of that? Now I'm trying to think. but at any rate, things naturally sort themselves out by density when you do this kind of stuff. I guess it's more normal for the sandy stuff to end up at the bottom and the rocks to end up at the top. But at any rate, the DNA pieces separate by molecular weight. And so you can look at the different bands that are formed in this centrifuge tube, and they're all made of nucleic acids, right? DNA or RNA or something like that. But what would make some DNA end up in one band and other bits of DNA end up in different bands? It's all DNA, right? So what's going on here? Well, there is when you do this with genomic DNA, there tends to be one primary band that is just the majority of the genomic DNA, but there are also smaller, what they call satellite bands, and the name is just coming from Latin. It means follower or courtier or bodyguard. That's all a satellite is. A satellite of Earth is just somebody, it's an object in space that follows the Earth like a bodyguard, I guess, in a sense. And so a satellite bit of DNA is just any DNA that comes out of this ultracentrifugation process that is separate from the main band of genomic DNA. So it turns out that the difference between animals with high C values, or i.e. lots of DNA in the nucleus, and animals with normal amounts, is repetitive DNA. So amphibians like salamanders don't exactly have way more genes than us, although some animals and plants do have more genes than us, but like generally speaking, these kind of other vertebrates don't have more genes than us, but they do have a lot more repetitive DNA. And this buildup of repetitive DNA is what expands the size of their genomes and gives them so much extra DNA in their cells. So it's not really that they're any smarter or any better than us. It's just that they have more, more bits of repetitive DNA floating around. By the way, if you're curious, how did they know that these satellite bits of DNA were repetitive without being able to sequence it in any kind of meaningful way? This is the 60s, so this would be a little bit before sequencing was like technically possible or technically feasible on any kind of real level. Sanger invented that, but it came a little bit later than 1961. It's kind of, I think it just bears mentioning because it's a little bit clever. You can tell how repetitive a bit of DNA is by quantifying the reassociation rate. So follow me here. If you have DNA, it'll naturally base pair with itself and form double-stranded DNA. That's just the natural order of things. That's the way DNA would prefer to be. It's the normal configuration. If you heat the DNA up, it'll eventually denature and break apart into single-stranded DNA. This is the essence of, or this is the first step of the PCR process, is you heat the DNA to 95 Celsius and it denatures and it breaks apart, and that allows you to bind primers to it and so forth. But so if you denature the DNA by heat and then allow it to reassociate under a very carefully controlled temperature gradient, you can quantify the amount of time it takes for the DNA to fully reassociate. Half the amount of time it takes to reassociate is defined as the C0T. That's this figure here. And to make a long story short, researchers discovered that these satellite DNA bands that came out of the ultracentrifuge had much quicker C0T values than did the genomic DNA. So it just turns out that it's easier for satellite DNA that's very repetitive to reassociate because the exact match doesn't have to be very precise. If you have DNA that's super repetitive, it can bind here, or it can bind here, or it can bind here, because it doesn't really matter where the repeats line up. They're all repetitive, so you don't have to get a one-to-one match. You can start here and then have the other one start halfway up the repeat length, and it'll still form a double-stranded base pair. So repetitive DNA reassociates much more quickly, and this is why researchers were able to tell that microsatellites had repetitive DNA in it. And this put everyone's fears to rest. We are better than amphibians because our DNA, though it has many repeats, it doesn't have as many as they do. So you can so just to summarize, you can genotype people on the basis of microsatellite repeat length. Microsatellites are spread throughout the genome in a fairly regular pattern, so you could find dozens or if not hundreds every chromosome. You can get there's enough of them out there that you can genotype widely without missing very much of the genome. So now that you have a marker or a signpost, you can somewhat find your way in the dark because you have something to genotype that tells you which haplotype a person got. So let's get over, let's go back to the topic of linkage one more time. So this is a review, and I'll bring this up a couple more times because I just think this is quite essential, and I think it's also a little tough to explain, or maybe I just have a tough time explaining it, but I think it's very essential to the concept of genetic studies in general, and I'll just take a number of tries at explaining it because it's just that critical. So during meiosis, there is a crossing over phase, and this is when homologous bits of mom and dad's chromosomes recombine to create novel chromosome combinations that don't exist in your own cells. So meiosis is a process that allows you to create new combinations of genes or of DNA that you don't have yourself, and that also have never existed on Earth before. And nearby stretches of DNA are more likely to be inherited together. Stretches of DNA that are distant from one another are less likely to be inherited together. In other words, for any two locations on the same chromosome, the closer they are, the less likely they are to undergo recombination. This is another word for this crossing over phase. It's called homologous recombination. So the tendency for nearby parts of the chromosome to be inherited together is called linkage. It's because it's as though they're linked in the heritability process. And linkage can be measured in centimorgans, which is a unit that people will still use today, but it roughly corresponds to a probability of what a centimorgan is. Basically, it defines the probability that two regions will be recombined during meiosis. So a centimorgan is when you have when the recombination rate between two locations in the genome is about 0.01 or about 1%. They're not literal measures of distance, but they're fairly proportional to distance, because regions that are very close to each other hardly ever recombine, and regions that are very, very far apart recombine almost all the time. So there is like a relationship with distance, but where recombination happens is usually centered around breakpoints in the genome that are repetitive, or that are like the same for everybody. So these breakpoints are the same for everybody on Earth, or at least everyone within a given population group. So recombination happens between those, not at totally random locations. So centimorgans aren't completely the same as distance in the genome, but they're fairly close. So let me just get into a little visual analogy explaining how distance and recombination rate are related. Now, I'll ask you to bear with me while I give kind of a little story, or like a little analogy, to help kind of make this go down a little smoother. But like, imagine you have a chromosome, say chromosome 3, and it has 200 million base pairs for its entire length. One scenario would be that you could break it into two equally sized fragments of 100 megabases each. If you did this, you could be very easy to take these two half chromosomes and put them back together in the correct order, because there's really no other way for them to go. You could put them backwards, but that would be very obvious to the cell that it's backwards, because there's so much sequence identity in each of those two halves. The 100, the half of the 100 megabases that is half of chromosome 3 is unique. It doesn't match anywhere else in the genome, so it can really only be put together back in the original order. Now imagine, so that's sort of what I'm depicting here, is a hypothetical scenario where you're crossing over half chromosomes. Now imagine another scenario where the DNA is broken up into 200 million single nucleotides, one base each. This would be too much fragmentation, because you could create infinite amounts of genetic variability, but you'd never be able to put the chromosome back together in the same order. A long stretch of DNA has a specific sequence identity and can be mapped to a very precise location in the genome. A single base has no identity whatsoever. It doesn't go anywhere in particular. If you have an A, it could go just about anywhere in the genome. There's all kinds of places in the genome that have an A in it. If you have an AT, or like a dimer, then at least you have something to work with. There's way fewer ATs than there are A's. 1/4 of the nucleotide bases in the genome are A's, or not exactly 1/4, but close to it. Way fewer are ATs. If you have a 3-mer or a 4-mer or some larger number of bases put together, the odds of seeing that any old place go down. It becomes more and more specific. So ultimately, the DNA is being broken up into fairly large-sized chunks to do this homologous recombination, because if it were broken up into too small of a chunk, it would be impossible to put the chromosomes back together again. So there's sort of an optimal length, I guess, for these recombination fragments that's based on the sequence identity. But this optimal length that we talk about is exactly the reason why proximity is so closely tied to linkage. If there's a certain size of recombination fragment that's optimal or biologically sort of the most common, then it would follow that any variants that are located within a distance of that size would tend to be linked, whereas any variants that are located a distance from each other that is further or bigger than that size would tend to not be linked. You see what I'm saying? So homologous fragments tend to be fairly long, and it follows from this that variants that are nearby on the same fragment tend to be inherited together. So here is an example of a linkage diagram where we've got the maternal chromosome in your body and the paternal chromosome, and we've got a couple of variants just highlighted here on the maternal chromosome just for fun. Pretend each of these triangles is a SNP. Well, when recombination takes place, the breakpoint is right here, and so one of the chromosomes inherits the first two variants from mom, and the other one inherits the second two variants, but they travel together. What really matters is these variants are traveling together. So Under these conditions, if you know the identity of this variant, you can guess with a fairly high level of accuracy that this variant, or that you would also be able to know this variant. Now let's look at this in the context of repeat length polymorphisms. This is the essence of a linkage study. So here, this is a repeat, a microsatellite or a short tandem repeat in mom's chromosome. And the breakpoint, we're going to keep it in the same place. So this repeat length polymorphism is going to be inherited. And alongside it, let's pretend this triangle is an actual disease-associated SNP or a disease-associated gene. So this repeat length variant is inherited alongside the disease gene. The reason they're inherited together is because they're linked. They're on the same haplotype. Here's another example of the same thing. Here is a disease-linked variant, but this time there's a short repeat length fragment. So that also gets inherited the same exact way. And it's linked because it's on the same haplotype. So what I'm trying to show you here is that if you have a disease-associated variant, it's on the same haplotype as a repeat length polymorphism. Because of linkage, you don't need to know what the disease SNP or what the disease-associated gene or variant is. All you have to know is what haplotype the person has. And you can look at that on the basis of repeat length polymorphism. You can genotype these repeats and you'll know where they got their haplotype from. So you can use this approach to infer the location of trait-associated genes. How do you do that though? Well, let's look. I would describe these repeat length markers as like signposts in the genome. These repeat length polymorphisms are spread throughout the genome fairly widely. You could, if you wanted to look, you'd find hundreds of them. They're all over the place. And what's important is that you can design a panel and genotype them in ways where you would capture most of the linkage in the genome without having to genotype very much of the genome altogether. Now, it's important to point out again that these markers they don't have anything to do with the trait themselves necessarily. Repeat length polymorphism is a form of human genetic variation that is assumed to not really have an impact on phenotype. It's just sort of random variation that exists within the population. Now, as we'll learn when we talk about autism spectrum disorders, that's becoming increasingly, or people are starting to think that actually might not be true, that repeat length variation might actually be a pretty important thing. But as far as we're talking about here, we assume that it's not really too important. So anyways, these are variants that you can genotype easily. They're spread throughout the genome and in linkage with most of the other, most other parts of the genome are in linkage with one of these repeat length markers, and they can be easily genotyped. And they vary considerably within the population. So there's enough polymorphism that you don't need a huge number of people to get a very diverse set of repeat length polymorphisms. So a linkage study is going to work on the assumption that trait-associated loci and markers are, a linkage study is working on the assumption that trait-associated loci, SNPs and other kinds of variants, and markers like repeat length polymorphisms are traveling in close proximity. So if you take a family tree where there's a higher rate of a given disorder, and you follow the disorder through the family, and this is a typical pedigree plot. So you would have the filled shapes are people who have the disease, and the empty shapes aren't, and the circles are females, and the squares are males, and all the rest of it. You can follow the disease through a family tree, and then what you do is you simply follow the repeat length markers through the very same family tree. So you would take all of the people in this pedigree and you would genotype them for a whole panel of repeat length markers. And then you would look, okay, what are the repeat length markers that are there in all of the cases, but not there in all of the non-cases or all of the controls. And by doing that, you can build up a sense of which parts of each chromosome seem to travel with the disease. This gives you an idea of where the disease causing variants are located on the chromosomes, even though you have no idea what actually is even there. All you have are these signposts that tell you, oh, there's probably something in this area. So let's look at this plot a little more closely. This is kind of a fake it's a cartoon example, but imagine you've genotyped families for A, B, C, and D. These are repeat length polymorphisms, let's say. So you could either have, so there's a variety of different alleles. You could either have A, B, C, or D, and you're just genotyping for them. So in this first pair, you've got a case that has the A and B alleles. And then you have another person who's not a case and they have the C, D alleles. Okay, so can't really tell very much from that. But then if you look at their kids, you have another case who has the A allele. They got the D allele from their mom and the A allele from their dad. And then the other person has 2 copies of the C allele. Then you look down one more generation and then you see another case and they have the A and the C alleles. And then you have 2 more cases. They also have A and C. One more generation down, another case, you've got A and C. And then down here, you've got another couple of cases, A, C, and AD. So if you look at this, and this is very simplified, it seems like anyone who's a case has the A allele, and the other ones, B, C, and D, can be there or not, and it doesn't really matter. But if you happen to have the A allele, it means that you have a likelihood of having the disease. Imagine doing this genome-wide where you're looking at 100 or so different markers for every person. What this would tell you is that whatever genes or whatever variants happen to be in linkage with the A allele, those are the ones that are likely associated with the disease. This is before the genomics era. You don't know what genes those happen to be. All you know is that wherever it is in the chromosome that the A allele lives, anything in that area is a suspect for causing the disease. Do you see what I'm saying? So you're genotyping things that are signposts. They don't tell you what's there. They just say, over here, this is where something might be happening. Because whenever somebody has this marker, they also might have the genes that cause the disorder. It's really that simple. So you can do a whole bunch of complicated math on this. These studies are very sort of troublesome to run because the genotyping process is very cumbersome, and you have to go through large pedigrees, large family trees. You can't just pick random people from the population. You have to have a known pattern of relationship and inheritance. But anyways, you get a measure called LOD. It's roughly the likelihood of linkage between a given trait and a given locus. So this is a type of study that is rarely done these days. This is a very old-fashioned kind of study that's been since replaced with the GWAS designs, with the GWAS designs that we'll be mostly talking about. The big advantage to using a GWAS design is that you can look at the the disease-associated markers or traits, the disease-associated variants themselves. You can actually look at the SNPs instead of inferring that there are SNPs. And also, you don't need to have a related population. There are some conditions that are rare enough that it's very tough to get detailed pedigrees for these conditions. It's hard to get a family tree of people who suffer, say, from some kind of rare condition. Schizophrenia is actually reasonably doable because 1% is fairly high as far as prevalence, and it's also very heritable, so you can find families where there's a lot of it going on. But linkage studies also have some advantages, including a known population frequency. You know a lot about the population you're studying in a linkage study, so there are certain assumptions you don't have to make that you normally do with GWAS studies. So hopefully that made sense to you. I think it's kind of what's funny about these older fashioned methods is, to me at least, they're much harder to understand than the modern methods. A GWAS study in its essence is quite simple. You measure all of the variants you can, you put people into 2 piles. Do they have the disease or do they not? And then you just look to see if any of the variants correlate with your likelihood of having a disease. And it's a very simple design. If I were to, it's really no different than saying, okay, well, we're going to put everyone into 2 piles in this classroom, and we're going to look to see if your hand size correlates with your skull shape or something like that. Like, you can make correlations between any kind of different measure, and the GWAS studies essentially use that logic, whereas the linkage studies, you have to understand what linkage is, and you have to understand these sort of more complicated experimental designs involving family trees and pedigrees and whatnot. So ironically, the older methods are sometimes more confusing. And yeah, to be honest, like, when you look at what used to be needed to do genetics and genomics work, the level of effort required at the lab bench compared to now is astonishing. It's really a wonder any of this was ever done in the first place, because it just is so labor intensive. But I'm speaking as somebody who's been deeply spoiled by the ability to just program everything on the computer and just type code in all day instead of having to get in front of the lab bench and pipette stuff and run gels and whatnot. So the earlier linkage studies that were done on schizophrenia did not really reveal anything too interesting. So this is an example. This is sort of like a Manhattan plot where you're showing chromosomal locations all the way from chromosome 1 to 22. Again, they mostly don't include the X chromosome or the Y chromosome on these kinds of studies. And then on the Y-axis of this plot, they're showing the negative log 10 transformed statistical significance. So the higher the number means that given polymorphism or that given marker is more significantly associated with schizophrenia. And the red points are from a sort of global analysis, and then the blue dots are from studies conducted only in Europeans. So the cutoffs for genome-wide significance are given here, and the cutoff for genome-wide suggestive, which is not statistically significant, but it's so, it's close to being significant, so people want to keep the results, are here. And what they find basically is in these like large meta-analyses is that even though vast amounts of work have been done trying to use linkage studies to understand schizophrenia, very little is actually concrete enough to replicate. Very little of it replicates. In fact, nothing here in this meta-analysis conducted by Ng et al. In 2009 reaches the level of genome-wide significance. So many, many years of linkage studies have produced essentially nothing of use for the concept of schizophrenia, even though it's very clearly, or like very arguably, a genetic condition. Now, this isn't to say that the linkage studies are useless. There have been a number of important hits over the years. BRCA1, which is a breast cancer-associated gene discovered by Mary Claire King in 1990, is a famous one. You will be tested for BRCA1 if you happen to be somebody with breasts, or like most likely you will be at least. And this is quite predictive of breast cancer, and it was discovered by linkage studies. So they're not useless, but I guess for something as complicated as mental illness, they're really not going to deliver the goods. You have to go for more modern methods. But as long as we're talking about older methods, I wanted to take you down a little bit of a pathway to learn about DISC-1. Now, you might recall DISC-1 from your first assignment. I put it in there, but maybe you didn't know as much about it when I asked you, but now I'm going to try to teach you a little bit about what DISC-1 is all about. So the earlier studies done on the genetics of psychiatric illnesses would tend to focus on pedigrees or on families with a high rate of mental illness. So in 1990, St. Clair et al. Published a study of a Scottish family with a high rate of schizophrenia, schizoaffective disorder, and recurrent major depression. So a family where there's a lot of serious mental illness running in the family. And by doing old-fashioned cytogenetics, where you actually look at the chromosomes and you look to see if all the bands are in the right place, again, incredibly laborious stuff, much further beyond where I'd ever want to be. You get to be very spoiled by just programming computers all day. But anyway, what they found was that this family had a balanced translocation from 1 to 11, from chromosomes 1 to 11, that was much more common among the family members that happened to have the mental illness than those without. So 16 out of 34 members of this pedigree had this translocation and some kind of mental illness, whereas those without mental illnesses, only 5 out of 43 had the translocation. So this is a pedigree chart. Don't worry about this too much, but it just shows you that these things are really used in real life. They have the diagnoses going all the way back several generations and to the current generation, and then they have marked out whether or not they possess this translocation. So this is a hint that something important might be happening somewhere on chromosome 1. So in case you hadn't heard, a balanced translocation happens when pieces of chromosomes get mixed up. In a balanced translocation, this is when they trade places, more or less. So what's happening here is a section of chromosome 1 breaks off and reattaches itself to chromosome 11, while at the same time, a corresponding piece of chromosome 11 breaks off and reattaches it to where the piece on chromosome 1 was. So you have two pieces of chromosomes just trading places. This is an important mechanism for evolution, but in everyday life, it's actually almost always harmful. You really can't have pieces of chromosomes getting mixed up in this way. It looks like this a little bit. So you have your normal chromosome set up, and then for whatever reason, a piece breaks off one and another at the same time, and they simply swap places. Now, they don't exactly know what's going on here, but the suggestion is that these parts of the chromosomes, because this translocation is a fairly major change, these parts of the chromosomes must somehow be causing mental illness, causing these schizophrenia type conditions. Now, I should point out that like these translocations are not ideal. You wouldn't want one if you could avoid it. But assuming the break point isn't over an important gene, you might get away without too many problems, because your cells don't really care what chromosome a gene is on. The chromosomal mapping of genes is almost totally arbitrary, with the exception of the sex chromosomes. What really matters is the genes have to be complete. So if you took a chunk of chromosome 1 and put it onto chromosome 11, as long as the chunk is complete and you haven't broken across the middle of any genes, you might get off without any real problems. Down syndrome, so that's in general, but of course that's not what happens most of the time. Translocations are typically fairly harmful, either to health or to reproduction. It becomes it can become difficult to reproduce successfully if you have these translocations. So Down syndrome can be caused, maybe about 5% of Down syndrome cases are caused by a translocation of the long arm of chromosome 21 onto the long arm of chromosome 14. So if you recall, Down syndrome is normally caused by having an extra an extra 21st chromosome. So you're normally supposed to have 2 copies of every chromosome. If you have trisomy 21, you have a 3rd copy. And so the actual pathology of Down syndrome is caused by having extra copies of all the genes on chromosome 21. But this isn't the only way it can happen. What matters is having extra copies of those genes. You can also have a translocation that doesn't give you an entire extra 21st chromosome, but it just takes a bit of the 21st chromosome and sticks it onto the end of chromosome 14. Ultimately, you'll now have 2 copies of chromosome 21 that are normal, plus an extra half copy of it grafted onto the end of chromosome 14. The net result is a higher gene dosage. You've got more copies of those genes on the long arm of chromosome 21 than you normally would, and this causes the Down syndrome phenotype, even though you technically you don't have an extra 21st chromosome, you just have an extra piece of it that's floating around there. There are also intersex conditions. For example, you can have XX males. This happens when the SRY gene, and we'll learn about SRY in our lecture on sex at the end of the year, is translocated onto the X chromosome. So we'll hear more about that down the road. But you can have conditions where you have 2 X chromosomes but are effectively male in terms of your phenotype and even in terms of your genetics, because SRY is the only gene on the X chromosome on the Y chromosome that really makes a difference for that kind of stuff. And of course, various cancers can be caused by translocations as well. And as I said, the act of translocating a chromosome isn't itself that bad. Intrinsically, you can rearrange the chromosomes however you like. What matters is if you break up genes or give yourself extra copies of a gene. You really can't have extra copies of a gene. Dosage control is the main thing when it comes to genes. Think about X inactivation is a big effort to control the dose of X genes, and the same is true for a lot of other conditions as well. But so translocation isn't that bad on its own. Cells don't really care where genes are. But if the breakpoint happens to span across a gene or something else that's important, you can have problems. So in 2000, Miller et al. Hypothesized that this illness that was reported by St. Clair et al. Was because there must be an important gene on chromosome 1 that was disrupted by the translocation. If there's some kind of important gene there and you mess it up with translocation, that could explain why that particular variant or that particular form of variation is so closely tied to schizophrenia and other such conditions. So, in 2000, Miller et al. Reported on the discovery of a gene on chromosome 1 that spans this breakpoint. They gave it the name DISC1, and DISC1 stands for Disrupted in Schizophrenia 1. So it's one of those things where you've discovered a gene, it's the early 2000s, Bill Clinton's still president and everyone's feeling fine. And, 9/11 hasn't happened. You're on top of the world. Everything's great. It's the end of history. And you're thinking, we've discovered the gene for schizophrenia, folks. I found it by looking at this weird Scottish family, and I'm going to name it the disrupted in schizophrenia 1 gene because I've the first gene for schizophrenia. And I'm kind of being a bit facetious there, but there was a bit of triumphalism. I suppose many of you may not have been around for this, but there was a lot of triumphalism around the turn of the millennium, right at the era kind of between 1999 and about 2000, and basically between 99 and before 9/11, everyone really sort of felt like things were going great, and the world had basically, we're on our way to fixing all the major problems in the world. And so it's the kind of era when somebody would discover something like this and give it such a triumphal name. But anyway, they discovered it, and they had no idea what it did or what the purpose it served was. But it was, as a fairly major discovery because it was the first gene you could concretely link to schizophrenia. It was a major breakthrough. It really was. And this was just kind of around the era when genomics was entering the field. The human genome was about to be sequenced, or the draft of the human genome sequence was about to be released. So it was a pretty important thing. They, and I think it kind of broke a bit of the insecurity that researchers had. So researchers, as I had been mentioning, there was a lot of skepticism about psychiatry and about the psychiatric medical model for mental illnesses that had been forming since the probably since the 60s, when there was a cultural reaction to all forms of coercive authoritarian control, and psychiatry was seen as a form of coercive authoritarian control. So there was certainly a pushback against it from the more libertarian segments of the population, as well as at the time sort of more of the left-wing segments of the population, who felt that it was really just a tool of coercion to suppress or to induce conformity. And people doing these kinds of studies did feel a little bit insecure, because while it seemed very clear that things like schizophrenia were major illnesses that were heritable and must be genetic, if you asked them where the genes were, they couldn't tell you, or what the genes even did. So this discovery of a schizophrenia-associated gene really broke the curse, I guess you could say. And they finally thought, okay, finally we found some kind of genetic thing that's associated with schizophrenia. Schizophrenia really is a real thing. It's not made up. It's not some kind of Freudian delusion. It really is a real thing with a real biology to it. Everyone felt a great sense of relief in those days. Now, this is before they really knew what DISC-1 did, but it inspired everybody to go to work trying to understand what this gene actually did. It was a brand new gene. Nobody had a clue what it did. So a variety of studies were done where they looked at the expression of the DISC-1 protein, and they found, Kirkpatrick et al. In 2006 found, that it was mostly located in the dendritic spines of healthy human brains. So this is turned out to be a type of cytoskeletal or scaffolding protein involved with governing neurite outgrowth. So the growth and development of little neurites, these little projections from developing neurons that could either form axons or dendrites. They make connections with other neurons. This is where you would see DISC-1 expression. And that's kind of nice because synapses are where all the good stuff in the brain happens. This is sort of where all of the action is. So if you find out that this gene that you know is linked to schizophrenia is located in synapses, you have a wonderful story you can tell about what might be going on. Researchers created a whole bunch of DISC-1 knockout mice. These mice had reductions in neurite outgrowth during cortical development and reduced dendritic spine complexity and cortical volume. So this is great. You've got a gene that you know now is expressed in human dendritic spines and is involved in the cytoskeleton during neurite outgrowth and the development of the brain. When you knock it out in mice, you interfere with those very exact processes, causing problems. You cause them to have shriveled up, unhealthy-looking dendrites and fewer synapses and whatnot. You also see deficits in spatial memory and working memory, the Morris-Water maze and these kinds of tests in mice. And ultimately, you end up with a number of behavioral and neuroanatomical markers that are very reminiscent of what you see in schizophrenia. Here, by the way, is an electron micrograph of DISC-1 immunoreactivity. So they've labeled DISC-1 in a section of the human brain, and you can see these dark colored areas as places where there's labeling. I don't really know a whole lot about how to read those electron micrographs, but I'm told this is a labeled terminal. Here is a dendritic spine that's been labeled, a postsynaptic density, a PSD, that's also been labeled. So there's a number of synaptic, anatomical, histological structures that are labeled with DISC1 in the human brain. And if you knock out DISC1 or somehow interfere with its function, you cause a bunch of schizophrenia-like conditions in mice. So that's perfect, right? What more could you ask for out of a gene? In 2008, Cavaggio and company created a mouse line that had a human-like DISC1 mutation. So they essentially created a version of mice that have a mutated copy of DISC1 that is very similar to what humans have with this breakpoint. So you have a deletion on exon 6 with 25 base pairs. You have 2 premature stop codons, exons 7 and 8, as well as a premature polyadenylation site. That stuff isn't too important. But what does matter is that when you make these mice, they produce a truncated DISC1 mRNA, or a truncated transcript, and therefore a non-functioning DISC1 protein. This is what you see in humans as well. They have truncated DISC1 transcripts and a non-functioning protein. And these mice have a number of working memory deficits. So they have all of the, as best we can tell by looking at mouse behavior, they have all of the mouse behavioral properties of schizophrenia, to the extent that a mouse could even have schizophrenia. And they also have anatomical deficits that are reminiscent of what you see in human brains. So here is some microscopic imaging of dendritic spines. You all remember what dendritic spines are. Here they are in wild types. All of these little points are dendritic spines, and you can see that the dendrites in these guys are very nice and spiny. This is in the dentate gyrus, which is part of the hippocampus. But the wild type normal mice have lots of nice little spines, but the mutants here, the homozygous mutants, they have way fewer spines, and here's the bar graph quantifying it. The mutants have significantly fewer dendritic spines in the dentate gyrus. And it turns out that if you were to look at brains from human schizophrenia patients, this is exactly what you'd see in their brains as well. So here's a control brain stained by the Golgi-Cox method. All of this line here would be a dendrite, and all these little dots are dendritic spines. The dendritic spines are where the synapses are located. And if you look at a dendrite in a human schizophrenia patient in the dentate gyrus, you'd see that it's very smooth. There are very few dendritic spines. So it's great, right? You've got this gene where if you mess it up the way that humans have it messed up, you get schizophrenia-like behaviors and brain phenotypes. Problem solved, right? We've nailed it. It's DISC1. It's disrupted in schizophrenia 1, and it causes all the things you'd expect. Well, not so fast. We have to ask a very simple question. DISC1 clearly is involved in the phenotype we see in this Scottish family. But that's just one family in Scotland. Does it show up in the general population? Schizophrenia makes up, about 1% of people in the world have schizophrenia. So you would expect this to be a very common mutation if it's what causes schizophrenia. You would, if the effect is as powerful as the data seem to say, you should be able to see this all over the place in the population. The surprising answer that began to emerge once we started doing genomics studies that were inclusive of the entire genome and looked at huge amounts of people, the surprising conclusion was that in the general population, there is not an association between variants in DISC1 and schizophrenia. In fact, there seems to be no association whatsoever. So here we have a meta-analysis done by Matheson et al. In 2012. This is a zoomed in Manhattan plot. So imagine if you take a Manhattan plot of the whole genome and you just look at one little area. So you've got the p-value of the association on the y-axis and the position along the chromosome on the x-axis. And right here is the DISC1 locus. They've highlighted it. And what you'll see is that there are no SNPs or no variants in the DISC1 locus that surpass the threshold for genome-wide significance in terms of the association with schizophrenia. What this means is that while DISC1 mutations caused by this translocation seem to cause schizophrenia in this one Scottish family, in the broader population, it doesn't. So this is a one-off case that applies only to a limited number of people. It is not a general explanation for how schizophrenia works. And in fact, as far as we can tell, the DISC1 gene as a whole, not even this one breakpoint, but the DISC1 gene as a whole seems to have really nothing to do with the inter-individual variation in schizophrenia. So that's awfully disappointing, isn't it, folks? You can't rely on biology to be the master link or to bolster a weak genetic link. DISC1 does have a lot to do with brain development. It does play some kind of role in forming synapses, and if you don't have it, you're in trouble. However, almost everyone has it, and it's not a problem for most everybody. So this is an example of a kind of false start in the world of psychiatric genetics. And in fact, we will find that schizophrenia is a polygenic disorder. Linkage studies were not able to find anything, nor were candidate gene studies. So in spite of the fact that we've established schizophrenia is a very, appears to be a very genetic condition, up until, as far as I've told you so far, nobody's found any genes that actually seem to cause it. So this is a kind of a big problem for the world, isn't it? Because we think there must be genes there, yet we haven't found them. Now, enter genome-wide association studies, GWAS studies. We've learned about how these work before, but what they allow you to do is take much, much larger sample sizes and scan the entire genome for anything that might be related to the condition that you're curious about. I should point out that you do still, so this is kind of just a power calculation, but you do need to have very large samples. So people have estimated that it would take about 860,000 participants to capture about half of the SNP-based heritability for schizophrenia. So it's very difficult to find that many people who have schizophrenia. So really there's a gold mine of information out there that we just can't access because we don't have enough participants even in the world to look at. But we're doing much better than we were in the past because genome-wide studies really do give you they really do deliver the goods. So here in 2014, we have a study published by the Psychiatric Genomics Consortium, the PGC. And this is their latest case-controlled GWAS meta-analysis on schizophrenia. So what they're doing essentially is they're just, every couple of years, they'll combine all of the genome-wide studies on schizophrenia and put all of the data together and see if when you add all those studies together, do you get enough, does the statistical power reach a point where you can discover new disease-associated variants? So Natalie asks, I think I missed it, but was the Matheson et al. Study a GWAS study? Let me go back to that slide. The Matheson et al. Study was a meta-analysis. So what they were doing was combining GWAS studies into one and looking to see if you took all the GWAS data available and put it together, would you be able to find an association with DISC1 then? People put these things together because, if you have a small sample size, there might be something really going on, but it might be beyond your ability to detect because the sample size is too small. So it's a meta-analysis of genome-wide studies. So here in this study done by the PGC, they're looking at 36,000, almost 37,000 cases and over 113,000 controls. And with that number of people, they found 108 schizophrenia-associated loci. But each locus had a very small effect. Even for the biggest effect loci, the odds ratio scores were less than 1.3. So we're talking about very, very small odds ratios here. Think about an odds ratio of 1 means you have a 1% of developing schizophrenia. You have the same odds as anyone else. A 1.3 odds ratio means that if you happen to have that variant, your odds go up to 1.3 instead of 1. That's not really, that's not a big increase in absolute terms, right? You're still talking about almost certainly, almost 99% chance you won't develop schizophrenia. So these are polygenic conditions, or this is a polygenic condition where a number of alleles all contribute a very tiny amount to the overall picture of heritability. Now here is the Manhattan plot from this study. Here they're looking, it's the same as any other Manhattan plot. We've got the chromosomes here along the x-axis, and we've got the significance on the y-axis. You can see the p-values get to be very low. This is one of the advantages of having a very big study, is you can get more and more power. But And there's a number of significant locations, right? All of these locations here with the little diamond-shaped points are significant. But what comes out on top is what's called the MHC locus. This is the major histocompatibility locus. This is a very complicated region on chromosome 6 that has many immune system genes. And this happens to have the strongest association with schizophrenia. So this turns out to be fairly important. I think that just looking ahead at the slides that I have, I may not get through everything that I wanted to tell you about this today, but don't worry, we've got 2 lectures to cover it, so nothing to worry about. So the major histocompatibility complex is located on chromosome 6, and it turns out to be a hotspot for schizophrenia-associated variants. This has been shown in a number of GWAS studies, and the more studies you do, the more powerful this association seems to be. The MHC was originally discovered in the context of tissue transplant compatibility. What used to happen is they would, back in the olden days, they would breed mice to the point where you could develop colonies of mice that were almost identical to each other in every way, except for tissue compatibility. You could try to transplant a liver from one mouse to the other, and it would be rejected. And through a variety of linkage studies and other methods, they determined that some part of chromosome 6 is where the variation that determines tissue compatibility is located. It turns out that what actually happens at this locus is this is where genes for MHC molecules are coded. And the major histocompatibility complex molecules are a part of the immune system. And basically what they do is they display fragments of self-derived and foreign peptides to the immune cells. So in your cells, there's a continual turnover of protein. Proteins are being synthesized. After a certain point, they'll be lysed or proteolized, and little fragments of peptides will be left over. They go through subsequent steps of metabolism, and they're eventually broken back down into their constituent amino acids. So there's this kind of continuous cycle between protein synthesis and proteolysis that breaks the proteins down. This happens for all of the proteins your cells naturally produce, but it also happens for foreign proteins, proteins that come in on bacteria or on viruses that aren't part of your body. These are also being not being synthesized, but they're being broken down by proteolysis. Now, the MHC molecules have the job of taking these little fragments created by proteolysis and pulling them up to the cell surface and just holding them up there for the T cells to see. So in a way, they're displaying little bits of random proteins they find floating around and saying, hey, look at this. And this is kind of important because it's kind of like, I don't know, it's kind of like a bit of a cellular lost and found. Like the cells are continually digging through the protein pieces they have floating around and holding them out and saying, hey, is anyone looking for this? Is anyone looking for this? Is anyone looking for this? The T cells are going around looking at this antigen presentation and saying, that's just part of me. Oh, that's just part of, that's part of this. Like they're going around trying to determine whether the fragments are part of your body, they should be there, or if they're foreign and need to be attacked. So the MHC molecules play a big role in governing the immune response because they display epitopes or pieces of proteins that are then assessed by your T cells as to whether they belong in your body or whether they don't. So it's a big region of the genome that controls this very critical immune function. The human MHC locus is on chromosome 6, and there are a whole bunch of MHC genes. There's MHC class 2, class 1, and class 3, all located like in this tightly packed region where it happens to be very polymorphic. The class 1 MHC molecules are expressed by every cell, and they present all kinds of random epitopes or protein fragments to the killer T cells to show off, and then the T cells will decide whether those things belong there or whether they don't. The class 2 ones are expressed mostly by professional immune cells like the macrophages and the B cells and the dendritic cells. So these are kind of closer to the action where infection is more likely to occur. So these are sort of a more sophisticated, a more sophisticated form of the overall process. And the class 3 molecules, we won't worry about those, but they also play a role in immune function. So this region on chromosome 6 has a lot of immune things happening on it. And this is very interesting to discover as far as the connection to schizophrenia goes, because there's clearly an association between schizophrenia and inflammation. And by inflammation, I mean immune activation. So, for example, DeNicola and company back in 2013 found that if you look at the circulating blood, cytokine levels in the blood of people with first episode psychosis, so the initial presentation of schizophrenia, they have higher levels of a number of inflammatory cytokines. You also see a differential expression of MHC genes as well as a number of other immune genes in the brains of people with schizophrenia. So here is one example of that. This is the levels of MHC1 protein in the prefrontal cortex, or the dorsolateral prefrontal cortex. And what you will see is the levels of this protein are lower in the brains of people with schizophrenia compared to the levels in the control people, the control brains. And so there is this like connection between inflammation and schizophrenia that's very attractive. And if you have a gene that's part of the immune system and thus plays an essential role in inflammation that is so tightly associated with schizophrenia, you have this wonderful possibility for a connection between immune-brain interactions and the disease itself. So I only have 2 more slides left. Maybe I'll just keep going. This is a taped lecture, so if you have to leave and go to your next class, that's fine. You can just come back and watch the last 5 minutes afterward. I apologize. I'll try not to go long as often, but it's just hard to resist getting more and more content in to tell you about. But anyway, the major histocompatibility complex molecules are expressed in the brain, or rather they're expressed throughout the brain. And you can find them located in the axons and the dendrites, as well as at glutamate synapses and astrocytes and microglia as well. So this was a bit of a surprise to researchers working in the sort of mid-2000s, because they had assumed for a long time that the brain was immune privileged. The immune system didn't have to do anything in the brain because the brain had the blood-brain barrier protecting it. It had its own kind of resident immune cells like the microglia, but they just assumed that there wasn't very much immune stuff happening in the brain. And it turns out that that's precisely the opposite of what's really going on. There's a ton of immune stuff going on in the brain, and the immune system is very important in just regulating the actual development of the brain. But this was all, fairly new knowledge at the time. So the major histocompatibility complex class 1 molecules play a role in axon outgrowth and synapse formation. So if you make mice that are lacking MHC class 1 molecules, they're knockouts, they have an increase in synaptic density in the visual cortex. So here are the wild type mice. VGLUT1 is a marker of glutamatergic synapses, and you see that there's a certain number of these on the dendrites of the visual cortical neurons. And then if you look at VGLUT1 staining in the knockout mice, you see much higher levels of staining, meaning that there are more glutamatergic synapses when you knock out the MHC complex in mice. They also seem to mediate the effect of maternal immune activation. So here, maternal immune activation is a model that tries to mimic what maternal infection is in human beings. So you use lipopolysaccharide or something, some other kind of injection to activate the immune system of a pregnant mouse. And what you'll find is that the offspring of that mouse who had its immune system activated, those offspring have behavioral phenotypes that are similar to schizophrenia and other types of psychosis. So it turns out that if you, it seems that the MHC class 1 molecules mediate this interaction. So if you look at maternal immune interaction, you will see that it causes, or maternal immune activation, I should say. Maternal immune activation causes an increase in MHC1 expression in the mouse cortical synapses. So that's what this staining here is here. They've stained for the major histocompatibility complex, and you can see all these marks in the control mice. But you can see considerable more staining in the mice, the offspring of maternal immune activated mice. So these mice have a behavioral phenotype that's reminiscent of schizophrenia, and they also have much higher expression of the MHC1 complex. At the same time, once they've matured to adulthood, they have lower synaptic density. So a simple suggestion here is that the major histocompatibility complex is involved in synaptic pruning. And synaptic pruning, if you haven't heard, is mediated by, in part, the immune system and the microglia. So it could be the case that overstimulating the immune system basically achieves, by way of this major histocompatibility complex, an overly zealous pruning of the synapses in the brain. This immune-mediated pruning leads to reduced synaptic density in people who are either exposed to maternal infection or who have genetic variants that cause this thing to be more active in the brain. So this is a wonderful kind of interpretation that links together genetic variations in immune genes with the ongoing process of brain development, which, as we are increasingly finding out, is mediated by the immune system. So you have this brain-immune interaction that plays out during development that may also play a role in the genetics of schizophrenia, and may also play a role in understanding why it is that maternal infection, think back to our developmental programming lecture, seems to cause or seems to increase your risk of schizophrenia. So to kind of wrap things up, we have a long history in genetics with schizophrenia, and most of what I've talked to you about, think of DISC-1 and linkage studies and whatnot, is really a story about technological limitations and false starts. We thought we had something, and then it turned out to really not be what we were thinking. It wasn't what we were hoping it to be. However, we do know at a baseline that there are anatomical differences in the brains of people with schizophrenia. Altered synapse density in cortical and hippocampal neurons is a key marker. And genetic variants that cause variation in these synapse densities are easy to, they're conceptually simple to link to schizophrenia, because this is already what we know happens in the brains of people with schizophrenia. DISC-1 was really thought of as a great savior because it did cause, it did seem to regulate synaptic development, and everyone had high hopes for it. But the MHC complex, which has very strong genome-wide data, is another such example that seems to regulate brain development. And I think it's, that's kind of seems to be where the action is these days. The brain is a very complicated organ, and it requires the coordinated work of many, many different genes all working together to develop properly. So small variations that cause what you might consider to be small differences in the genome can actually have a fairly large effect on the brain, because its development is so complicated. It's not like it's not a simple thing. The tiniest differences can snowball into very large anatomical and phenotypic differences once you play out the brain development through your entire lifespan. So I'll leave you with one question to think about, which is the fact that schizophrenia is common enough in the population that it's a little bit tough to understand why variants that cause it, or that seem to play a role in it, even exist in the gene pool. It's clearly not adapted to have schizophrenia. So why is it that evolution allows such variants to hang around? There is, I'll just leave it and say, balancing selection is one hypothesis. It would imply basically that schizophrenia-associated variants have both positive and negative effects on phenotypes. So there might be trade-offs with variants that cause schizophrenia. Maybe the immune things help you in some way, but also hurt you in another way. Things that are balanced that way are very difficult to select out by natural selection, because the positive and negative effects kind of cancel each other out. Anyway, we'll leave it there, folks. Thanks for your patience. I hope that was interesting to you. We'll talk on Wednesday we'll talk more about the genetics and epigenetics of schizophrenia and what we're doing to try to find out what's going on with this condition. And hopefully show you, in as much detail as I can, the latest information that we have in this regard. So thanks to everyone for listening. As always, I hope you learned something, and we shall see you next time here on everyone's favorite class, Neuro 3301. And have a good week also.